Hi students, am I audible? Yes, am I audible to you all? Hi everyone. Hello. Hi everyone. I hope you all are doing well. First of all, let me know, am I audible and visible to you all? Yes. Good evening everyone. Good evening. Please tell me, am I audible and visible to you all? Okay, so welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. So I was supposed to start my class at 6 p.m. but your HSP sir was teaching you, I think, nucleophilic addition reaction, if I'm not wrong. It was nucleophilic addition reaction or something. I don't know the topic exactly. So that's why I started it late, right? Sorry for the inconvenience. Okay. Now, because we have started the class late and we need to finish the mutations, disorders and the pedigree analysis first. So let's complete these topics today. Okay. And then we will keep one shot of principles of inheritance and variation soon. Fair enough? Yes? Fair enough? Okay, so what you have to do now, you just need to send the link of the session, right? You need to share this link with your friends so that you can tell them that finally class has started. And even yesterday I was trying to take your class, but I don't know what was the glitch. Okay, so I was missing you. Literally, I was missing you. What about you? Are you done with the part that we have completed, principles of inheritance and variation? Whatever we have completed so far, are you done with it? Are you practicing the MCQs or not? Don't worry, soon I'm planning to take your MCQ revision sessions as well. Don't worry about that. Okay? Okay. So welcome. So quickly share the link with others. And now let's start the class. But before that, see, this is the amazing video I have posted today. Actually, I was about to post it 2 days back, but because of some glitch we were not able to. So do check out this video. Okay, it is having the proper planning. Even if you are the beginner, what you need to start for the biology, first thing. Secondly, for the physics as well, for the chemistry as well. Okay, for the physics as well as for the chemistry, right? You are going to get proper schedule here. You will get to know about the important topics there. Fine. So, Surya Bache, because this week we were not able to start your planned human physiology. Next week we are going to continue and I will increase the frequency of the classes also. So, we will finish the planned physiology as well. So, any other doubt, guys? Any other doubt? Sure. So, you know what you have to do. Just go and after the class, of course, just go and watch this video. Okay. So, see, all the detail is given here about the exam. Okay, I hope you guys have seen, and moreover, see the physics syllabus, right, which will help you to get 120 to 140 marks as well. So everything is shared here. Okay, and yes, I want to see your comments in the comment section as well. Okay, so now let's start the topic, and you know which topic are we studying. We are talking about the principles of inheritance and variation, and I know you guys are waiting for, right, you people are waiting for your one shot, but let's finish the pedigree analysis, mutation and everything first. Okay, done, bachi? So how many of you find it difficult? Yes, how many of you find it difficult, right, this pedigree analysis? Tell me, how many of you find it difficult ki ma'am, this chapter is actually very difficult? Yes, that this chapter is difficult, this topic is difficult, ma'am, we don't know anything about it, or something. Shiva Kumar, that will be there soon, don't worry. Easy it is, na? In today's video we will complete the disorders as well. We are going to cover everything in today's video, don't worry. We are going to start with the mutations first. See what we have covered so far. Let's have a quick revision of it. We started with the Mendel. Okay, father of genetics, Mendel, right? Here we have completed the laws of Mendel. Even we have discussed the gene interaction as well. You know about the gene interaction, right? Allelic and the non-allelic inheritance, right? Gene interaction, allelic and non-allelic interaction, everything we have covered here in this chapter. And other than that, right, because it is the part of this only, your pleiotropy, polygenic inheritance, right? These are the main topics that we have covered so far. Fine. And after it, we started with the chromosomal theory of inheritance. Yes, chromosomal theory of inheritance. So that's what we have completed so far, isn't it? That's what we have completed so far. Okay, and then we talked about the linkage and the sex determination. So if you want to study these topics, right, bache, so you can check the previous parts of this particular chapter. Okay, so today we are going to start our story from the mutations. So first of all, you tell me what is mutation. Quick, as per you, what is mutation? What do you know about mutation? Bala, I'm better now. Thank you so much for asking. What do you know about the mutation? Quick, tell me quickly. What do you know about the mutation? Quick. Okay, what happened? Just a minute. Just a minute, guys. I don't know what happened. Let me restart it. So yes, what is mutation? Tell me quickly. You answer it and then we will continue. Don't worry. It will start. Sudden changes. What else you know about the mutation? Tell me. What else you know about the mutation? Tell me. Alteration in the nucleic acid sequence. Okay, fair enough. What else? What else? Variation in gene. Some genetic changes in chromosome. Okay. And see, first of all, when you talk about the mutation, yes, you are right, they are the sudden changes. All of a sudden, these mutation will occur. And Hugo de Vries, right? Hugo de Vries, okay, he was the one, yes, Hugo de Vries, he was the one who noticed these mutations. Okay, who was the first one? Hugo de Vries, right? So that's what we need to study. I don't know what happened to the screen all of a sudden. It's okay, let's continue. So see, so mutation is sudden, inheritable. Each and every word is important here. They are no doubt sudden, inheritable. They can pass from one generation to another, but they are the discontinuous variation in organism due to change in chromosomes and the genes. So when you are talking about the mutation, right, it's not only the change. It is not just the change in the chromosomes only. It is also the change in the genes. Okay, it is also the change in the genes. Like for an example, you know that your DNA, right? Your DNA will coil. You know that we have the histone proteins. So DNA is going to coil over the histone protein. DNA is going to coil over the histone proteins, isn't it? And slowly, you know that it will form nucleosome. Further coiling will be there. Further coiling will be there. And finally, you are going to get your chromosome, isn't it? Isn't it? So, nucleoid, your DNA will coil over the histone proteins. It is going to coil over the histone proteins. It will form nucleosomes. Nucleosomes will further condense, right? Further, there are the models of coiling. And then finally, you are going to get your condensed chromosome, right? So, when it comes to the changes in the chromosomes, like there can be the deletion, there can be the inversion, there can be the addition. These are the words that we are going to discuss. And now when you are talking about the gene, the changes in the gene, now you know your DNA. The basic unit of inheritance here is the gene, right? And of course, here you are going to talk about the sequences, the nitrogenous bases, the nucleotides, of course. So here in this case, you will talk about the point mutations. You are going to talk about the frame shift mutations. So these are the words that we need to study here. Are you getting it? So when it comes to the mutation, what are they? What are they? So they are sudden, inheritable, discontinuous variation it is. Okay, what is it? It is the discontinuous variation. Okay, it is the discontinuous variation. And yes, when their when there are alterations in the genes, in the DNA, of course it will change genotype as well as phenotype. Understood? It is going to change genotype as well as, yes, it is going to change genotype as well as phenotype. Understood? Understood? So who gave it? Hugo de Vries. Clear, bache? Right? Now, the word here is discontinuous variation. Anyone here in the class who can explain it, that what is the meaning of this discontinuous variation? What is the meaning of this discontinuous variation? Anyone here in the class who can explain it? Quick. Discontinuous variation. Yes, Akansha, Sahi thi, Suhashini. Guys, what happened? Like for genetics, you were so excited, and these days I'm not even getting the response from your side. What is happening? Right? Not interested in studying biology? Not interested in studying biology? Done with biology? Or you are thinking that we will start it in last 3 months or 4 months? So it is not going to happen. Trust me. Right? No doubt you have to pay attention on physics as well as on chemistry, but with biology. Without biology, it is not going to help. Okay, so biology is like you have to study it every day. You have to revise it every day. Right? Even if you are not studying a new chapter, at least you should go for the revision. Sometimes examples, right? Sometimes examples, sometimes theory, sometimes that short note, summary, right? The digestive juices, the hormones, right? Morphology examples, diversity examples. So these are the things that you need to do. Okay. Okay. So now here the word is discontinuous variation. Why do I emphasize each and every word? Right? Why? Because bache, such questions will help you to answer assertion and reasons, one thing, and secondly statement based question. See, now I will relate this part with the evolution. Now when we talk about the evolution, I hope you know about the Darwin as well, Charles Darwin. Right? So as per Darwin, right, Darwin talked about the variations, and as per Hugo de Vries, he talked about the mutations. Darwin talked about the variations, Hugo de Vries talked about the mutations. So these mutations are discontinuous variation. Actually, as per Darwin, right, from parents to offspring, right, you know that the information will pass from parents to offspring, from parents to offsprings. Basically, generation to generation information is going to pass. So as per Darwin, there are some variations. They will accumulate with time, right? Slowly, they are going to accumulate. Like in our generation, let's say 2 variations are there. I'm explaining it in a very layman language so that you can understand. Okay, let's say I am acquiring 2 variations. In next generation, 4 variations are there. In next generation, right, 10 variations are there, 20 variations are there. So ultimately, there comes a day when these variations, they will be too much that they can produce a new species. Fine, that they can form a new species. It is as per Darwin when he was explaining the evolution, when he was explaining the speciation, that is formation of species. As per Darwin, when it comes to the evolution, these are the gradual changes, right? Slow changes that are going to accumulate with time. They're the slow changes that are going to accumulate with the time, and then you will see a new species. It's more like that. Now, when you talk about Hugo de Vries, if you can relate it with the evolution, he has given the saltation theory, right? Single step, large mutation, right? As per him, that's all of a sudden some changes will be there and they can form new species. Okay, so mutations are not like something which will pass from every generation to generation to generation to generation, not like that. All of a sudden some changes will occur, and that changes can go to the next generation. That's why no doubt they are causing the variations, but in a discontinuous manner. It is not from each and every generation, right? But in a discontinuous manner they are present. So that is the meaning here. Is it clear? Is it clear? Understand the genetics. We are not going to cram the genetics. We are going to understand the genetics. Is that clear? Okay. So now let's talk about the types. So here, when you are talking about the types, genomatic mutations are there, chromosomal aberrations are there, gene mutations are there, right? There are 3 things that we are studying here. What are we studying here, students? One is genomatic, second is chromosomal aberration, third is gene mutation. One is genomatic, second is chromosomal aberration, third is gene mutation. Now, genomatic and chromosomal aberration, these 2, right? Genomatic and chromosomal, genomatic and chromosomal aberration. Both are the part of chromosomal mutation, right? Both are considered as chromosomal mutations. Do you know that? Yes. And when you talk about the gene mutation, of course, here you are going to talk about the gene only. Clear? You are going to talk about the gene only. I know many of you, they don't know about the genomatic mutation. Wait, I will explain it. So when the word is genomatic, it means there is a change in the chromosome number. Which mutations are we going to consider here? The mutations which are causing a change in the chromosome number. I'll give you one example here. Now, in the case of humans, how many chromosomes are there? Students, I want you people to be interactive. Right? I want you guys to be interactive in the biology class, because it is very interesting. The subject is very interesting. And I know even teacher is not boring. Teacher is also interesting. Okay, so I don't want such responses that you are just, watching the class and you are like, interact. Please interact. Right? Although on YouTube, that is the basic thing. Teacher will come, teacher will teach, and teacher will go. That's how we go for the online teaching, right? But here it is not like that. It should be two-way. If I'm asking one question, you should answer it. Okay, even if you are going to ask a valid question, I will answer it. Fine? Fine? So can I see some good emojis here in the chat section? I don't want a dull class. I really don't want a dull class. Quick, everyone, quick. And yes, yesterday I learned something from my students. Okay, that is Tamil. Okay, you can correct me if I'm wrong. That is Vanakkam. Vanakkam. Irukinga, something Irukinga. I forgot that. Right, that if you want to start the class, so you should be like Vanakkam. One word is there, I just forgot it. And then Irukinga. Okay, that welcome to the class or something. How are you? And it is for how are you? Isn't it? Epidi Irukinga. Epidi Irukinga. Yepidi or Epidi. That's Tamil, na? That's Tamil. Okay, so let me start again. Okay. So, Vanakkam, Epidi Irukinga. I'm your biology educator Ambika Sharma, and this is what we are discussing. The principles of inheritance and variation, and right now we are talking about the mutation. And today finally we'll finish this chapter, and next week we are going to start molecular basis of inheritance. Okay, okay. Then Epidi Irukinga. That's what I have learned. It means how are you? And it means how are you? Next is Telugu. Next is then Kannada. So I will learn these basic lines first. Fine. Telugu, Kannada. This is what I'm going to learn. Okay. So now come here. So we are talking about the chromosomal mutation. So we were talking about the humans. So humans are having how many chromosomes? 46 chromosomes are there. Fine. 46 chromosomes are there. Now let's say if there is any change in the chromosome number, if it is 47, if it is 46, or if it is 45, okay, such type of mutations are genomatic mutations. What type of mutations are they? Genomatic mutations. And remember, polyploidy, aneuploidy. So polyploidy, aneuploidy, here the chromosome number is changed. No, so that is the part of genomatic mutation. Okay, that is the part of genomatic mutation. Clear? Now, chromosomal aberration. See, bache, aberration. What's that? That is chromosomal aberration. So it occurs due to alteration in chromosome structure or in sequence of genes arrangement. So basically it is the chromosomal aberration, right? There is some problem with the structure of the chromosome. Let's say here you have one chromosome, okay, a part of it is deleted. If that part is deleted, means that genes, that chromosomes are also deleted, of course. Or maybe there can be the addition. There can be something extra from another chromosome as well. So addition, deletion, inversion, right? Mutations like that, they come under the chromosomal aberration. And when it is gene mutation, as I said, due to the change in the chemical structure of genes, right? So it is at molecular level. Fine, bache. So Ashini, it is at molecular level. Here you will be talking about the point mutation and frame shift mutation. Point mutation and frame shift mutation. So mutations we will also study in the next chapter. Fine. Few things we will also study in the next chapter. So this is what mutations are. Is that clear? Is that clear? Clear, bache? Sure. So first time, as I said, first time Hugo de Vries, he started it. Okay. And can you tell me the name of the plant? Can you tell me the name of the plant? He was studying evening primrose. A very famous question, but yeah, it is there in evolution. Okay, so that's your Oinothera lemachiana. Oinothera lemachiana. Fine, that's the botanical name of that particular plant. And this plant is having the chromosomal aberrations. Fine, this plant is having the chromosomal aberration. Fine, bache. Sure, 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 sure. Okay. Sure. And even, you know, mutations are useful as well if I talk about them. They are mostly recessive, right? They are mostly recessive. And nature used to accept useful mutations, not the harmful one. But you know, exceptions can be there, okay? So, other than that, if I'll give you a very interesting example of beetle and totem. Do you know about beetle and totem? Beetle and totem, they conducted experiment on neurospora. Do you know what is neurospora, bache? Neurospora is also known as Drosophila of plant kingdom. Right? It is also known as Drosophila of plant kingdom. Actually, it's a fungus, right? But it is used for that biochemical studies. Your neurospora. It is a fungus, but it is used for that biochemical studies. So, a beetle and totem, they conducted experiment, right? One neurospora was the normal one, the wild type one. Another is the mutant one. So, when they cause changes in the neurospora, the mutant neurospora, right? So that is known as auxotroph. That is known as auxotroph. Are you getting it? Clear, bache? So that is what mutations are. Is that clear? That is what mutations are. Is that clear? Sure, bache? Sure, bache? So, you know, even many scientists, when they understand that how, you know, these mutations can be generated, right? How can we induce them? Okay, when they studied about it, so they used it in a positive way also, right? Like in the case of plants, sometimes in the plants we induce the mutations and we used to get a proper useful result for the human being. So this is about your, yes, this is about your mutation. So any doubt? Yes, students, any doubt? Sure? Any doubt here? Okay. Okay, done. So that's what mutations are. So this is what we are going to study as of now. So agents that cause mutation, that's your homework. Agents that cause mutations, they are known as mutagens. Okay, they are known as mutagens. So they can be physical, they can be chemical, they can be biological. So that part we will discuss in the molecular basis of inheritance. Fine. So now let's read it from NCERT, and then we will categorize, we will divide it into parts. So I know you people are free today, okay? So let's finish this portion nicely. Let's discuss the pedigree analysis. Let's discuss these disorders, because for sure 2 questions are going to come from today's lecture. So 2 question means 8 marks, right, Shiva? 2 questions means 8 marks. Fine. So as you can see, it's a phenomena which results in alteration of the DNA sequence and consequently results in changes in the genotype as well as the phenotype. So obviously, if there is any alteration in my DNA, if the sequence was like this, and if I'm changing it, so it is changing my genotype as well as my phenotype. Obviously, if genotype is changed, definitely phenotype will also be changed. Okay, so mutation. So in addition to recombination, you know na, recombination, crossing over. So in addition to recombination, bache, mutations are also very important for the variations in DNA, right? For variations, these 2 things are responsible. So when you are making your notes, that's how you have to proceed. Variations is equals to mutation plus combination, and these variations are literally important. Why? These variations are literally important. Why? Because these variations, bache, why? Because these variations, yes, they are important for the survival, they are important for the adaptation. Clear, bache? Clear? Okay, so now see, basic things are given, that DNA runs continuously, right? It is going to form the chromatid, it will further be coiled, right, bache? So therefore deletion or gain of segments of DNA as a result in alteration in chromosome can be there. So see, since genes are known to be located on chromosome, alteration in chromosome results in abnormalities or aberration. That's what we have studied, isn't it? That's what we have studied. And chromosomal aberrations, right, when there are structural changes in the chromosomes, that is very common in the case of cancer cells. Okay, that is very common in the case of cancer cell. Okay, bache? And if there is a change in the single base pair, then it will be the point mutation. So now let's elaborate this part. Okay, let's elaborate this part. So you can also make this flow chart. Fine, you can also make this flow chart. So are you ready? Yes, Saiti, everyone. Are you people ready for it? Quick. Are you all ready for it? Sure. So first is that include, right, chromosomal mutation, that includes genomatic, also known as heteroploidy, and second is chromosomal aberrations. And then here comes the aneuploidy and polyploidy. This is what we need to study here. Aneuploidy and polyploidy. Is that clear? Yes. Ritigna, is that clear? Aneuploidy and the polyploidy. I will explain it too. Fine, I'm going to explain it too. And then comes, yes, bache, deletion, inversion, duplication. Deletion, inversion, and duplication. Deletion, inversion, and duplication. So that comes under the, right, that comes under the chromosomal aberration. Okay, that comes under the chromosomal aberration. So deletion is there, inversion, duplication, and yes, how can I forget translocation? That's my favorite type of mutation. Okay, translocation, that's my favorite type of mutation. Fine, fine. So gene mutation, also known as point mutation. Gene mutations, because gene is that basic unit now that is present, and then comes the point mutation. So here you know that substitution of a base will be there, substitution of a base will be there, and then it is the frame shift mutation. Okay, it is the frame shift mutation. So these different type of mutations, they cause different types of disorder. Fine, these different type of mutations, they cause different type of, yes, different type of disorders. I'll give you one example. When it comes to the substitution, you have the example of sickle cell anemia. Okay, you will be studying the example of sickle cell anemia. And when it is the frame shift mutation, you will be talking about the thalassemia. Okay, we are going to talk about what? We are going to talk about the thalassemia. Okay, even thalassemia is also there. Okay, is that clear? Sure. So you have chromosomal mutation, right? Then you have gene mutation, right? Then you have gene mutation. Clear? Right? So that's what you need to know. So can we start? Yes, flowchart done. Bache, please post your notes in the group as well. I want to see it. Okay, I really want to see it. So can we start with the heteroploidy first? Yes, that is your genomatic mutation. Can we start it with first? Or you want to start with the chromosomal aberrations? Whatever you say, we'll go accordingly. Right? And mutagens also we have to discuss, don't worry. So we'll go accordingly. What do you want to discuss first? So let's start with the genomatic mutations. Genomatic mutation. So let's start this part, bache. So first thing is your polyploidy. What is the meaning of polyploidy? Poly means many, right? Many, right? Here, when it is the polyploidy, you'll be the particular mutant organism will have many sets of chromosomes, or you can say that there will be a change, right? There will be a change in the sets of chromosome. Are you getting it? What is this polyploidy? There will be a change in the sets of chromosome. For an example, normally, you know that organisms are diploid. What are they? They are diploid, means two sets of chromosomes are there. That is 2n. That is 2n. One is from mother, another is from father. One is from mother, another is from father. Now let's say if it is triploid, right? It means 3 sets of chromosome. So when there is an addition of one set of chromosome, one extra set of chromosome, bache think na, that is known as your, yes bacha, quickly tell me, that is known as your polyploidy. Okay, that is known as your polyploidy. Sure, that is known as your polyploidy. So here there is a change in the sets of chromosomes. So ultimately this polyploidy, it comes under euploidy. Okay, so euploidy includes your diploidy, triploidy and polyploidy as well. So polyploidy mein there will be many sets of chromosome. So diploid individual 2n, triploid 3n. So polyploid, it can be anything, right? Individual can also be 6n, right? So sets are more, right Ajay? Sets are more. Clear? Just say we are humans, we know we have 2 sets, 23, but let's say one more set is added. Isn't it? It will be like this. So then humans will not stay humans. They will change into a different species, right? We are humans because we have these 46 chromosomes, okay? So we will change into a different individual, okay? Fine. So that's what you need to keep in your mind. So that is your polyploidy. And polyploidy is very common in the case of P for plants. P for polyploidy, P for plants. In the case of plants, you are going to see that polyploidy is very common. Can you tell me the reason about it? Why is it so? I hope you remember when we talk about the plant cell division, right? When we discuss plant division, plant mitosis, so we talk about one thing there, right? That they do not have the centriole, isn't it? They just have the pericentriolar material, but they do not have the centriole, right? See, when I teach you, I don't want to just translate the NCERT, okay? My motto is that you should understand the NCERT, okay? You should not cram it, you should understand it. So if you want to understand the NCERT biology, you should relate all the chapters. So sometimes we just limit our information to a particular chapter only, and we don't know how to relate one chapter with the another one. So in my classes, this is what you are going to see. If I'm teaching genetics, sometimes I will jump into the animal kingdom as well. Even I'm going to talk about the morphology as well. So be ready for that. Okay, be ready for that. So right now, no doubt we are talking about the genetics, and suddenly I started the cell division. Why? Because here that particular topic is related. So I am telling you that when we talk about the plant cell division, so plants, they do not have the centriole, isn't it? They do not have the centriole. In their case, when there is the formation of spindle apparatus, so you know that only pericentriolar material is there. Okay, you have pericentriolar material only. What you have there? You have pericentriolar material there. Okay, that will form the spindle apparatus. But because these centrioles, they are going to put it at a proper place. Basically, they will keep it intact. They are going to arrange that spindle apparatus. So plants, they do not have centriole. They just have pericentriolar material. So chromosomes are not aligned properly. So in the case of plants, it's very common that after division, you will see sometimes triploid, hexaploid plants. Okay, Vidisha, that's very common in the case of what? That is very common in the case of, yes, in the case of plants. Clear, bache? Clear, bache? Okay, done. So polyploids, which are having even number of sets, like tetraploid, right? 4 sets are there. Hexaploid. Okay, so the polyploids having, you know, having even number of sets, they are mostly, you will see that they are fertile. Okay, they are fertile, they reproduce, they form seeds, and that's how they grow. Like the wheat, right? The wheat that we used to eat, Triticum. Fine, the wheat that we used to eat, Triticum aestivum, you know that? That is also not the wild type. That is also a kind of mutant. That is basically the hexaploid. Are you aware of it? Triticum aestivum. It is not even diploid. It is also hexaploid. Clear, bache? It is also hexaploid. So polyploid plants with even number of sets, they are fertile, they will reproduce, they will form the seeds as well. Clear, bache? Clear, clear, clear? Sure, sure. So yes, you can even write it. Huh? So sometimes, you know, we even discuss that whether, no doubt here it is 2n diploid. 2n diploid. So it is showing the chromosome set as well. So we have 2 sets. So if it is triploid, 3 sets are there. How many sets are there? 3. I'll give you one example. Let's say 21. Let's say a plant is having 21 chromosomes after mutation. Initially it used to have 14 chromosomes. So can I say that one set is added? Can I say that one set is added? Yes, Rithigna. Can I say so, Suhashini, that one set is added? There is a plant having 14 chromosomes. Means 2n is equals to 14. So n will be 7. So if it polyploidy occur, the complete set will get added here. So it will become 21. It will become 21. Yes or no? Yes or no? So here x is 7. So here, right? So how many sets of chromosomes are added here? Just one set. One extra set is added here. Okay, so if I'm keeping x as number of sets of chromosomes that are added, here it will be 1 only. Only one set is added. Okay, and how many chromosomes are added? 7 chromosomes. Right? How many chromosomes are added? 7 chromosomes. Clear? And there is one more example of this polyploidy. Do you know about triticale? Yes. Do you know about triticale? It's a very common example and the important one as well. Triticale. Don't you know that? See, triticale, triticum. Okay, so that's wheat. And another is, this is from rye. Triticale, it is not. Triticum, that is your wheat. Right, that is your wheat. And this rye is from Sickle. It is a cereal, na? Rye is there. So when you are hybridizing this triticum and rye, so triticum is your hexaploid. If I'm not wrong, it is 42 chromosomes are there, and it will be your 14 chromosomes are there. Right. So triticum is hexaploid. Your rye is diploid. Right, your rye is diploid. So what are you getting in triticale? Do you know that? What are you getting in triticale? Can you tell me about that? Anyone? Can you tell me about that? What will you get in triticale? Yes? Right? See, you are going to pick up the sets. Let's say your triticale is 4x. Okay, then right, x is what? It is different, no? X is what here? What is the value of x here? It's easy. What is the value of x here? In both the cases. In both the cases, what is the value of X? That's easy. Be quick. Yes, Akanksha, Suhashini, Ritigna. The value of X, bache. This is X. This is X. What is X? Thank God. X is 7. It is 7. Here also it is 7. So if your triticale is 4x, means 7, 7 plus 7 plus 7 plus 7, 4 times, right? 7 into 4, 28. If you are adding 2 more sets here, then it will be fertile. So yeah, if you are adding basically 4 more sets there, then it will be fertile. So it's like that. So triticale, you should know about the triticale as well. So it is also the example of your polyploidy. Okay, so we further discuss polyploidy. Just sometimes, you know, 2 different plants, right? 2 different plants, their chromosomes are added. Sometimes same plants and extra set of chromosomes are added. Sometimes different sets of chromosomes are added. So that's also that's how it varies. Okay, that's how it varies, but that is not the part of discussion here. Fine. So triticale example is important to understand. Now aneuploidy is very important, and we will discuss the disorders on the basis of aneuploidy. Fine. So if you understand these mutations, that disorders will be easy for you then. Okay, the disorders are going to be easy for you then. Okay, so aneuploidy. Here, bache, there is no change in the sets of chromosome. Instead of that, number of chromosome will change. Like here, if a plant is having 14 chromosome, if one set is added, it will become 21. If another set is added, it will become 28. Like this. So sets, addition of sets of chromosome is there, right? To this diploid organism, one set, another set, another set, another set can add. That's how it goes, right? Fine. So here there is the addition of the sets of chromosome. But here you are not talking about the complete set. You are talking about the number of chromosome. When there is the change in the number of chromosome. Let's say we are humans. We have 46 chromosome. If one extra chromosome is added, we will become 47. So that's the example of aneuploidy. If one chromosome is deleted, then total number of chromosomes will be 45. That is the example of aneuploidy. So aneuploidy is very important. Okay, aneuploidy is very important. So it is the loss as well as the addition of the chromosome, right? It is the loss or addition of chromosomes. Loss or addition of chromosomes in a set of chromosome, of course. In a set of chromosome, of course. Okay, bache, that's aneuploidy. Fine. That is what aneuploidy. So addition can occur and deletion can occur. There are 2 possibilities. Addition of a particular chromosome can occur. Even the deletion of a particular chromosome can occur. So can you give me some example? Can you give me some examples? Right? If there is a loss, if there is a deletion, if there is a loss, if there is a deletion, can you just give me the example? So instead of deletion, you should write it loss. That will be better. So bache, here you are going to talk about the monosomy. What will you discuss? Monosomy. Mono means single, right? If from the diploid set, one chromosome is deleted, that is your monosomy. That is your monosomy. Okay, and if there is the addition of one chromosome, here you are not talking about the set, that's why it is the trisomy. Best example to study the trisomy is trisomy of chromosome number 21. That is your Down syndrome. Remember, it is given in NCERT, right? Down syndrome. So it is the trisomy 21. What is it? It is the trisomy 21. It means there is an addition of the 21st, extra copy of 21st chromosome is there. Okay, extra copy of 21st chromosome is there. Okay, that is the trisomy and that is the monosomy. Monosomy, can you give me one example? Hope you remember Turner's syndrome. Turner's syndrome. Deletion of X. Deletion of X. Right? Turner's syndrome. Exactly. Fine. Exactly. Okay. So, trisomy and monosomy, these examples are given in your NCERT. But if it is the nullisomy, what is nullisomy? It's 2n minus 2. 2 chromosomes are deleted. Now, let's say if you are talking about the nullisomy in the case of humans, that particular human will have 44 chromosomes. That particular human will have 44 chromosomes. And here it is tetrasomy. Right? If there is the addition of 2 chromosomes, 2n plus 2, 2 extra chromosomes are there. Okay? 2 extra chromosomes are there. Clear, bache? Clear, bache? And sometimes it can be double trisomy. It can be double trisomy. Double trisomy means it can be 2n minus 1 minus 1. Do you know the meaning of double trisomy? Oh, it is trisomy, so there will be the addition, sorry. 2n plus 1, 2n plus 1. Do you know the meaning here? Double trisomy. Which double trisomy means here you are talking about 2 different chromosomes. Right? It is not mandatory that always there will be the addition of only 1 chromosome. Like 21st chromosome, extra copy of 21st chromosome is added. No. It can be 15th, it can be 21st as well. That's how it varies. Okay, that's how it varies. And here when it is the loss, it can also be double monosomy. So double monosomy means 2 different, right? There is a deletion of 1 extra copy, but that is a different chromosome. Clear, bache? Clear, bache? So addition of 1 chromosome in each set, right? In each set. Okay. Okay. So do you know what is the main cause for this aneuploidy? Anyone in the class? Anyone in the class? What is the cause of aneuploidy? What is the cause of aneuploidy? Anyone in the class? Of course, bache, non-disjunction. Okay. Non-disjunction of chromosomes. Non-disjunction of chromosomes. That is your aneuploidy. That is your aneuploidy. Fine. That is non-disjunction of chromosomes. Clear? Clear? So non-disjunction of chromosome during the meiosis. So here non-disjunction means, see, you know that when there is the anaphase, these chromosomes, they will split. Okay, or even if it is the anaphase 1 also. So these chromosomes, they are going to split this bivalent pair, right? It will get separated. So let's say if this is a particular cell, let's say if here non-disjunction occur, if these sets, they will not get separated, what is going to happen? Here you will be having 2 copies of this complete set, and here the another gamete will be having only 2 chromosomes. See, imagine after let's say after anaphase 1, right? Imagine it like this, that after anaphase 1, this is the scenario. This is the scenario. Usually we should take it in anaphase 2, that there is no proper separation of chromosome. Okay, so if it is anaphase 2, so you know that there will be the splitting of chromatid. Let's say 1 chromatid, it doesn't get split. Let's say 1 chromatid, it doesn't get split. So obviously this daughter cell will have 1 extra copy. Okay, it can have 1 extra copy. Fine. So that's how this aneuploidy occurs. So it is because of the chromosomal non-disjunction, means your chromosomes are failed to separate during meiosis. Fine. It's failed to separate during meiosis. Clear, bache? Clear, bache? So can we start some? Yes. Can we just start some examples? We should start some examples, right? We should start these disorders. or firstly let's finish the mutations only. Okay, let's finish the mutations only and then I'll give you the examples. Fine, then I will give you the examples. But first of all, tell me one thing. Now I'm starting chromosomal aberrations. Okay, that's a very common question that I'm going to ask. So chromosomal aberrations means there are structural changes. Okay, what is there? Structural changes are there. Chromosomal aberrations. Chromosomal aberrations. So what is there? Structural changes are there. Fine, structural changes. So you have to tell me one thing. Let's say you have a submetacentric chromosome, short arm and long arm. Which side is known as the short, what is the another name for the short arm? What is the another name for the short arm of the chromosomes? Short arm of the chromosomes. That's P or Q? G Vachin Avansh. That's P or Q. Short arm I'm asking. Short arm is P or Q. Very good. So when it is the short arm is P, and in such chromosomes, long arm is Q. Long arm is Q and the short arm is P. Sure? Short arm is P. So first point clear. First point clear. Now let's start the discussion of these chromosomal aberration. So let's start with the deletion. So deletion, as the name is saying, there is a deletion of the part of the chromosome. There is a deletion of the part of the chromosome. Let me quote one example. Okay. Okay. So let's say this is your chromosome. Okay. This is your chromosome. It contains some gene. Obviously, chromosome means DNA, DNA means sequence. So imagine it like this. So it is A, B, C, D, E, F, G, H, right? This is the sequence which is present on the chromosome. Are you getting it? This is the sequence which is present on the chromosome. Present on the chromosome. So A, B, C, D, E, F, G, H. This is the sequence. Now let's say when it is the deletion, might be this part will get deleted. Might be this part will get deleted. Right? So basically, if this particular portion is deleted, so this chromosome will miss the sequence A and B. This chromosome will miss the sequence A and B. Okay. So in this particular chromosome having sequence A, B, C, D, E, F, G, H. So if there is the deletion of A and B, so that chromosome will contain C, D, E, F, G, H only. Right? C, D, E, F, G, H only. Right? Right? So this is the deletion. So if ends are getting deleted, then you will use the word terminal deletion. Then you will use the word terminal deletion. So let's say if instead of GH, EF will get deleted. It is not mandatory that always ends will get deleted. Even, right, the part within, right, even this part within the chromosome can also get deleted. So obviously then you will use the word intercalary deletion. Right, intercalary deletion. What will be there? Intercalary. Intercalary is in between. Okay, in between. Clear, bache? Clear, bache? So here we read two words. This is extra. You want to write down, you can, or you can leave it. So it can be paracentric also. It can be pericentric also. So this is extra. It can be paracentric also, or it can be pericentric also. It can be paracentric also. It can be pericentric also. So do you know the difference here, paracentric and pericentric? Yes? Okay, so this is, no, this will not come under the deletion. This will come under the inversion. So I thought, yes, in the deletion also sometimes we consider this partner. No, centromere will be there. So that's all. This is all about the deletion here. So you will be having one is your terminal one, another is your intercalary one. Is that clear? I'll give you one example. Note down. Cry, do, chat, syndrome. Do you know about it? That's important. Please note down. Cry, do, chat, syndrome. It is the example of deletion. Do you know that? Cry, do, chat, syndrome. It is the example of deletion. Wait, let me write it. properly. So next part that we are discussing, it is the chromosomal aberration. Okay, it is the chromosomal aberration. So when the word is aberration, means there are structural changes, there is change in shape and the structure. So deletion is the first one that we are discussing. Fine. So this deletion can be terminal if ends are deleted. It can be intercalary if ends are not there, but you know the part within. So I gave you one example, and your example is, yes, your example is cry do chat syndrome. Which example is there, bache? Cry do chat syndrome. And here in this particular syndrome, there is the deletion of the short arm. There is the deletion of short arm, that is P, है ना? That is P of chromosome 5. Okay, there is the deletion of short arm of chromosome chromosome 5. Okay, short arm means P. So it is cry do chat syndrome, and you know in this particular syndrome that baby will have the baby will produce the sound like a cat. Okay, so this example is important. Cry do chat syndrome. Okay, it is cry do chat syndrome. Okay, cry do chat syndrome. Clear bache? Clear bache? Sure? Are you sure? Right? So this is deletion. Next part is, okay, inversion. Fine, inversion. So what is the meaning of inversion? You know na, inversion, inverted, inversion, inverted. Okay. Okay. Fine. G bache Roshan. So let's say this is your chromosome. This chromosome is having a sequence 1, 2, 3, 4, 5, 6. Right? And let's say if it will get inverted. If the sequence will become 1, 3, 2, 4, 5, 6. So normally it was 1, 2, 3, 4, 5, 6. But let's say if it become 1, 3, 2. So 2 and 3, they are inverted. At the place of 2, 3 is there. At the place of 3, 2 is there. So obviously it is changing the sequence. If it is changing the sequence, accordingly protein formation will also change, right? Accordingly the phenotype will also change. So this is the inversion. So inversion can be of two types. One is paracentric and another is pericentric, right? One is paracentric, another is pericentric. This is extra. Okay, paracentric means when centromere is not included. Centromere is not included. It is not included. Like see this example. Okay, so this part will get inverted. There is no involvement of centromere here. But if it is pericentric, I means include, right? I means include. This is how I remember. I means include. So centromere is included. Centromere is included. This is a simple way to remember it, right? This is a simple way to remember it. You are right, bache, right? When it is the inversion, there is the breakage of chromosome, there is the reunion, but you have to understand the literal meaning first, then the topic will be easy. Inversion, opposite. So obviously if 3 will come to the 2's, if 3 will come to 2's place and 2 will go to 3's place, so obviously firstly there is the breakage of chromosome, then the joining will be there. Right, breaking of chromosome and then the joining will be there. So this is the inversion. The example is not given in NCERT, but that's your homework. You will check the example and you will mention it in the comment section. Okay, you are going to mention it in the comment section. Clear bache? Clear, clear, clear. Sure? Okay, sure? Sure. So in our syllabus, the example, this cry do chat and even the example of translocation is very important. These two examples are very important if you talk about your syllabus. Next is duplication. So now let's say if this is the sequence A, B, C, D, E and F. If it will get duplicated, if it will become A, B, C, D, E, F. Of course, it is also not normal, isn't it? It is also not normal. Duplication is also not normal. Yes or no? Yes or no? So here can you see that, right? No doubt I am saying that the part here, one part, this AB is duplicated. You have two AB now, right? So obviously here, can I say that this duplication is also somewhere the addition of chromosome? It is also the addition of chromosome. Can I say so? AB to this AB, a part of AB got added. Okay, so this is the duplication. So when it comes to the duplication, the example is bar I character of Drosophila. Bar I character of Drosophila. Actually, bache, in Drosophila, if X chromosome, right, if there is a if this X chromosome, if the segment of X chromosome, if that segment got duplicated, so in the case of Drosophila, instead of normal eyes, you know, some narrow eyes will form. Okay, some narrow eyes will form. Clear, bache? So that is the duplication. So bar I in Drosophila. Okay, so there is a duplication of a gene which is present on the X chromosome. So that gene is actually the B gene. That gene is actually the B gene. So that is the duplication. Clear? That is the duplication. Fine, bache. Then comes translocation. Translocation is important for your syllabus, right? We discuss the examples of reciprocal translocation as well. See, again, the this word is explaining everything. Translocate, like this is one chromosome, this is another chromosome. A part of this chromosome, it is getting attached to this chromosome. It will get translocated from this part to this part, isn't it? So there will be deletion of the original chromosome. See, this is that type of mutation, and here there is the addition. There is the addition. I'm using different colors. I hope it is clear to you. I hope it is clear to you. So just look at it. Just look at it. It is a one chromosome. Let's say chromosome A. This is chromosome B. So a part of chromosome A, it will move from this chromosome A to chromosome B. So it will translocate now from this part to this part, from this chromosome to this chromosome, from this chromosome to this chromosome. It will translocate. So obviously the first chromosome, the A, it will become shorter, isn't it? If you talk about A, can you say that? Deletion is there, of course. If you talk about B, can you say that? Addition is there. Can you see that? Addition is there. This is translocation. Okay, this is translocation. Fine, 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 fine. Right? So sometimes, you know, the crossing over, usually it occurs in the homologous pair of chromosome. Crossing over, it takes place in the homologous pair of chromosome. But let's say crossing over is there in the non-homologous pair of chromosome. Okay, crossing over is there in the non-homologous pair of chromosome. Just imagine, just imagine. So in that case, this thing can happen. Okay, okay, right? This thing can happen. So I'll give you the example of one disorder. This is important. Philadelphia chromosome. Right, Philadelphia chromosome or chromosome or CML, right, or it is known as CML, Philadelphia chromosome of chromosome number 22, chromosome number 9, okay, or it is known as CML. Do you know CML? Yes. Do you know CML? What is CML? One is Philadelphia chromosome or CML. What is CML? CML is your chronic myeloid leukemia. Fine. So here there is a translocation. This is important, bache. Here there is a translocation in between chromosome 22 and chromosome 9. Okay, so it is the translocation in between chromosome number 22 and chromosome number 9. Clear, bache? Right, a segment from chromosome 22, it will move to chromosome 9. So chromosome 22 will be your Philadelphia chromosome. Clear? Chromosome 22 will be your Philadelphia chromosome. Clear, bache? Clear, bache? Sure, 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 sure. So CML Philadelphia chromosome, CML chronic myeloid leukemia it is. And yes, let me tell you, see, now this is one example where I'm saying a part of A will join with part of B. But sometimes, you know what happened? There is a reciprocal translocation also. Like the part of this B will also join to this A. The part of B will also join to this A. So it is reciprocal translocation. It is reciprocal translocation. Like from this, there are two chromosomes. If a part of it is moving to this one, so the part of it is moving to this one. So reciprocal translocation is there. So Philadelphia chromosome, right, your chronic myeloid leukemia, this is the example of reciprocal translocation. Is that clear? This is the example of reciprocal translocation. Is that clear? Sure? Is that clear? Done, bache? So it can occur if there is an exchange of segments in between the non-homologous pair of chromosomes. Because when it comes to the crossing over, this is what we used to say. In crossing over, this is what we used to say, that we have a pair of homologous chromosome and the non-sister chromatids of that pair. See, here you have what? You have pair of homologous chromosomes right, homologous chromosome. So what is happening here, bache? You know that the non-sister chromatids of homologous pair of chromosome, they exchange the segments. But sometimes what happen, bache? Right, if there is the crossing over in between two non-homologous pair of chromosomes, that can result in the translocation. Okay, that can result in the translocation. So that's what you need to keep in your mind. Fine, that's what you need to keep in your mind. Sure, bache? Yes, sure. Okay, so next is gene mutation. Uh-oh. So in gene mutation, you have the example of substitution, okay, and another is your point mutation. Just a minute, guys. Okay, done. So power cut was there, so now it's fine. Okay, fine. So next is substitution and point mutation. Next is substitution and point mutation. So bache, substitution. To substitute at the place of A, if you are putting B, that is the substitution. If at place of A you are putting B, what's that? That is substitution. Clear? Substitution, to substitute. So I hope you remember sickle cell anemia. Today we will discuss it in detail. Right? I hope you remember sickle cell anemia. When your RBC, it will become sickle shaped. And your sickle cell anemia is the example of pleiotropy, multiple phenotypic effects of a gene. Your sickle cell anemia is also the example of codominance, where that defective allele and the normal allele, they are, right? They are simultaneously, they are dominating themselves. Okay, so sickle cell anemia is the example of substitution. How? Because I told you in beta chain of hemoglobin at 6th position, in beta chain of hemoglobin at 6th position, right? Your glutamic acid is replaced by valine. Okay, glutamic acid is replaced by valine. So we have a trick. Trick is good valine. Okay, good valine, good glutamic acid. So don't write glutamin here. It's glutamic acid. It's glutamic acid. Is that clear? It's glutamic acid. So glutamic acid is glutamic acid, it should be frame shift mutation. Wait, I'll explain. So glutamic acid is replaced by valine. Clear? Good valine. So glutamic acid is coded by GUG. Valine is coded by, okay, glutamic acid is coded by GAG. Valine is coded by GUG. This is actually the triplet based codon. Triplet based codon which is forming that particular amino acid. We will talk about it when we will start molecular basis of inheritance. So substitution, sickle cell anemia at, in B chain at 6th position, glutamic acid will be replaced by valine. Glutamic acid will be replaced by valine. Okay, so GAG will become GUG. GAG will become GUG. Clear? So GAG will become GUG. So if you look at it very carefully, at place of A, that is your adenine, U will come. Okay, there is a change in the one base only. There is a change in the one base only. So that's why we also call it as point mutation. So gene mutations are also known as point mutations. Clear, bache? So this is your substitution. The sickle cell anemia is the best example of substitution. Okay, the sickle cell anemia is the best example of substitution. Clear? So this substitution can have two types. One is transition and another is, one is transition and another is transversion here. Okay, one is transition, another is transversion here. One is transition, another is what? Another is the transversion here. Now, transition. Here, bache, purines replaced by, I know I should explain it after molecular basis of inheritance, but there also I will explain it again, so don't worry about it. Right? Don't worry about it. And pyrimidines, they are replaced by pyrimidines only. Now, when you talk about the DNA structure, see, this part even I have explained in the chapter biomolecules. Right? In the chapter biomolecules also I have explained it, that we have nitrogenous bases. One trick is PUGA, another trick is cut. PUGA, PU means purines. And here these are your pyrimidines. Fine, these are your pyrimidines. So purines include guanine and adenine. Pyrimidines include cytosine, uracil and thymine. So cytosine and thymine is in DNA. Cytosine and uracil is in RNA. So if in the substitution at place of, let's say, adenine, guanine is there. Or if at place of guanine, adenine is there. So this is the example of substitution. But what type of substitution is there? Transition. Okay, transition. Clear? Clear, clear, clear. It is the transition. Okay, and when, so yes, transversion means if at place of purines, like purine is replaced by pyrimidine or vice versa. At place of pyrimidine, purine is there, or at place of purine, pyrimidine is there, at place of pyrimidine, purine is there. So it's more like that. So this is the example of substitution, transition and transversion. Fine, transition and transversion. Is that clear? Is that clear? So next to it is your frame shift mutation. Right? Next to it is your frame shift mutation. Okay? So frame shift, here it includes a addition or deletion. Like let's say, this is your, I'll give you a very basic example. Let's say when it is your DNA. Right now I'm not talking about the mRNA. Let's say if it is your DNA, it is not your mRNA. Let's say. Now, you know that from DNA mRNA will form. From DNA mRNA will form. So basically this mRNA will form the protein. This is what we know. DNA forms mRNA, mRNA forms protein. This is the flow of information, that is, this is a central dogma of information. So DNA to mRNA is your transcription. What is it? It is your transcription. mRNA to protein is your translation. It is your translation, right? So DNA to mRNA, transcription, mRNA to protein, that's going to be your translation. That's going to be your translation. Now my point is, if there is any change in the DNA, of course that change will reflect in mRNA, of course that change will reflect in protein, in that enzyme, and it will change our phenotype as well. Okay, it will change our phenotype as well, isn't it? Yes, isn't it? Of course, yes. Of course, yes. So now let's say if there is a mRNA, that mRNA is having the sequence, certain sequence is there. Right, so from mRNA protein will form. So we consider 3 bases. 3 bases will form 1 amino acid. 3 bases will form 1 amino acid. So let's say if there is the deletion of one extra, if there is the deletion of one extra base here, let's say initially it was AUG, UUU, CCC, GGU. So if let's say there is the addition of G here, one extra G. So first is AUG, then it will become GUU, then it will become UCC, CGG. So frame of reading will change. So Hashini, frame of reading will change. Moreover, if there is a deletion, then also frame of reading will change. That is also frame shift mutation. Fine. So when we will finish molecular basis of inheritance, you will understand it in a better way. Okay, you will understand it in a better way. So all I can say is frame shift mutation, and where there is the deletion of the bases, right? The disorder is thalassemia that you will study in this chapter. Okay, it is thalassemia. Fine, it is thalassemia. So it is all about the mutation. I know we need to discuss the mutagens as well, but that we will do later. Fine, that we are going to do later. So we have to start the pedigree analysis now. See, you know mutations, you will understand some disorders. If you will understand pedigree analysis, you will understand the inheritance of that disorder. So both the topics are important. So let me finish this pedigree analysis part, and then we will start that sickle cell anemia, Down syndrome, Turner syndrome, this and that. So we'll finish it soon. Fine, we are going to finish it soon. And today let's finish this chapter. I thought that I will take the one shot of of this entire chapter, but it is not possible because I started the class late. Your HSP sir ended it late, so okay, that's fine. Even chemistry is important. Fine, even chemistry is important. Okay, okay. Fine. So let's go. So the next part that we have to discuss is pedigree analysis. This pedigree is not the this pedigree is not that dog food. Okay, it is an important topic. Fine, it is an important topic, and trust me, it's easy. They are not asking these days, they are not asking the question from this particular part. Okay, but still you cannot miss it because it is important, right? This portion is important, this pedigree analysis. Okay, and remind me, we have to discuss the sex limited and sex influenced traits as well. Okay, we need to discuss the sex limited and sex influenced traits as well. So first thing that we have to discuss is your pedigree analysis. Okay, it is pedigree analysis. Fine, pedigree analysis. Okay, so let's start. So it is basically going to tell you about the inheritance. Okay, just say you might have studied like you might know that sometimes we used to say that this particular character is in your family history. Just say, let's say if your grandmother is having some heart related issue, so yes, even you can consider that yes, you can also get heart related issues. Just say diabetes is very common. It is related to the genetics, right? That genes when they pass from one generation to another, right? You know it very well. Genes when they pass from one generation to another, this is how the genes are going to pass. No doubt there are variations, no doubt recombination is there, Mutations are also there without any doubt. Recombination is also there. Mutations are also there. But students, right? But if you will see the inheritance of a trait in a family, right, you can study that pattern actually. You can study that pattern actually. How? With the help of this pedigree analysis. And why? Why this pedigree analysis is important? Because we can study inheritance of certain characters. Okay? We can study that. We can link that. Fine. Just for an example, if I talk about this sickle cell anemia also, it is also autosomal recessive disorder. Fine. So you can study it in family history. So these pedigree analysis, they help in that. Okay? They help in that. Wait. See. So if you have to start the genetic disorder, first of all, you should know about it. So see, the idea that disorders are also inherited, right, has been prevailing in the human society since long. So this was based on the heritability of certain characteristics featured in families. In families, there is the inheritance of certain features. Of course, we know that. So after the rediscovery of Mendel's work, the practice of analyzing inheritance pattern of trait in human beings began. Means after the discovery of that Mendel's work, right, scientists, they started studying what? They started analyzing the inheritance pattern, that how these things are, you know, how these things are inheriting, the basic process behind inheritance. Okay, they have started studying that. So since it is evident that control crosses, it is evident that control crosses that can be performed in pea plant or some other organism, they are not possible in case of human beings. Study of the family history about inheritance of a particular trait can provide an alternative. So we cannot perform such crosses now that we were doing in the pea plant, like homozygous dominant, homozygous recessive, then their mating will be there. Of course not. Of course not. But here, this is what we can do, right? Family history can be studied. Right, that particular trait, its inheritance can be studied. So such an analysis of traits in several of generations of a family is pedigree analysis. Okay, such an analysis of traits in the several of the generations of family is actually the pedigree analysis. So in this analysis, the inheritance of a particular trait is represented in the family tree over generations. So that is what we are going to study here. We will study the inheritance of a particular trait. So in human genetics, pedigree study provides a strong tool, fine, which is utilized to trace the inheritance of a specific trait, some abnormalities or the disease, bache. So some of the important standard symbols will be used here, and then we will study that how that inheritance, you know, how that things will go, right? So some symbols will be used in that family tree. But before that, understand few things. One is autosomal dominant trait, like we divide such traits as well. Firstly, understand this, then we will talk about the symbols, right? Autosomal dominant traits, fine. Autosomal recessive traits, this is as per your syllabus. Sex linked, or you can also call it as X linked dominant trait, and then sex linked recessive traits are also there. Sex linked recessive traits are also there. And bache, you have Y linked inheritance as well. What do you have? You have Y linked inheritance as well. Clear? Clear? So that's what we have to study. Autosomal dominant, autosomal recessive, sex linked dominant, sex linked recessive, and Y linked inheritance is there, right? So Ashini, so first of all, understand this. What is the meaning of autosomal here, your autosomes here, and what is the meaning of allosomes here, right? What is the meaning of allosomes here? First of all, you know that we humans are having 23 pairs of chromosomes, isn't it? We humans, we are having 23 pair of chromosomes. Out of that, 22 pairs means total 44 chromosomes are the normal body chromosomes. Okay, normal body chromosomes. They are not going to define the sex of that individual. They are not linked to that. Okay, they are not linked to that. Clear, bache? Clear? And when it comes to the allosomes, in the allosomes you have just one pair. Like in the case of females, XX. In the case of Y, XY. So when you talk about the inheritance of the traits which are present on these autosome chromosomes, like let's say if one gene is present on your 20th chromosome, if one gene is present on your 20th chromosome, you are taking it a like autosomal inheritance, right? How are you going to take it? That it is the example of autosomal trait. It is the gene for that particular character is present on our autosomes, normal body chromosomes there. If let's say there is a gene which is present on the allosome, okay, allosome means sex chromosomes, that X and Y, okay, so it is related to that. Then you are talking about what, right? So then if any particular gene is present on the X chromosome, right, so that will come under your sex linked inheritance. Okay, sex linked inheritance. Clear bache? Clear bache? So now see, one word is dominant here, another word is recessive, right? One word is dominant here, another word is recessive. So bache, dominant, so you know that these genotypes are for the dominant trait, and when it is the recessive one, this is the genotype. Am I right? Dominant trait, capital A, capital A, capital A, small a. Recessive, right? Small a, small a. So if we want a recessive trait to express itself, both the alleles need to be recessive. Both the alleles, they need to be recessive, yes or no? So Ashini, Nandini, Adepu, yes, tell me. Quickly answer it. So if it is the dominant trait, this is the possible genotype, and if it is the recessive trait, this is the possible genotype. So it is autosomal dominant. Autosomal dominant trait means this will be the genotype, and this particular gene is located on autosomal chromosome. This is the genotype here, and here it is sex linked dominant and recessive. So dominant, recessive, clear. Dominant, recessive, clear. Now you have to study their inheritance. So look at these symbols here, right? You have to follow the NCERT for this. So bache, if it is a square, it is denoting male. Circle, female. If this is the shape, sex is unspecified. But if they are filled, like if let's say they are bubbled or filled, completely filled or bubbled, right? So it is showing affected individual. It is showing affected individual. Like if you are talking about a disorder, right, and the male, that box is colored, right, it is colored, so it is showing that individual, that male is having that particular trait, that particular character, and this sign here is showing the mating, right? Single line is showing the mating. So here you can see that the male, female, and here the mating is there. So, and if this pedigree tree is like this, so what is the meaning here? It means this is the parent generation, right? Father, mother, and this is their children. Two daughters are there. So this is the next generation. If let's say there are two lines, so it is showing the marriage in between relatives. Okay, let's say the marriage in between the same family, between the cousins. So it is consanguineous marriage. Okay, consanguineous mating. It is in between the relative. So see, parents above and children below. So in order of birth, left to right. So here they are showing parents normal, right? But the male child is affected here, and 5 unaffected offspring. So you need to remember these symbols. Sometimes question can directly come from these symbols. Okay, right? Directly from these symbols, question can come. Fine, bache. So now there are some points. We will discuss it and then we will move. So see, for quick revision, what is pedigree, genotype, phenotype? So here you guys can see autosomal trait that is located on autosome chromosome, non-sex chromosome. Sex-linked trait that is located on sex chromosomes. Homozygous, heterozygous, we have discussed already. Fine. So these are some extra symbols as well. But yes, you have to focus on the NCERT only. But and even this is important. It is showing twins, dizygotic twins, and they are monozygotic. This is important. Monozygotic, single zygote. Single zygote is, further dividing and forming further two embryos and fetus. So here the sex of the individual is going to be same. See, 2 sisters are there. Here they are dizygotic. These are the fraternal twins. Okay, two different zygotes are there. So this is also important. Fine, this is also important. And see, siblings, that's how they are showing. Fine, that's how they are showing. So it is as per NCERT. It is as per NCERT. Now, these are some rules. Fine, these are some rules. I always explain this topic with the help of these rules. Okay, okay. So the first point is dominant. Dominant. Whenever you talk about the dominant trait, this is what you need to keep in your mind. Okay, forget everything. If you will focus on these 2 points, na, you can answer all the questions. Fine, you can answer all the questions if you will focus on these 2 points. Whenever you talk about any of the dominant trait, the very important point is it does not skip generation. And on the other hand, when you will study the recessive trait, you will say that generations are skipped. This is, according to me, this is the most important point to rule out whether that particular inheritance pattern is of a dominant trait or the recessive trait. Okay, okay. This is a very important factor, and you can rule out which type of inheritance is there. Dominant, recessive, dominant, recessive, dominant, recessive. Now, doesn't skip generation or skip generation. What is the meaning of that? Here, if let's say there is any trait which is dominant. So, let's say if there is a trait which is dominant, right? I'm not making a proper inheritance pattern, but let me tell you. So, if any trait is dominant, if it is there in the parents, you will definitely see it in the next generation, in the children as well, and in the next generation as well. Okay, I'm not saying that all the individuals will be having that particular trait. They can have, but it will not skip generation. Let's say in next generation, you will also see that particular trait, or you will again say the particular trait, right? So ultimately generation is not skipped. In this generation also you will see that character, in next generation, and in next to next generation. In next to next generation that character will be there. So whenever it is a dominant trait, right? So if parents are having that disorder, definitely you will see it in their children. I'm not saying in both the children or in all the children's, but it will be there in the children. Next, if children are affected, now they will become parent, they will also inherit it to their next generation. So no generation will be skipped. No generation will be skipped when it is a dominant trait. So if parents are affected, definitely their children are. Or you can say that if children are affected, definitely their parents are also affected. One thing. Now when it is a recessive trait, recessive. Now, in their case, sometimes what happens, parents are totally fine, they are normal. Okay, parents are totally fine, they are normal. But still you will see that trait in the children. And sometimes parents are affected. Sometimes parents are affected, but children are normal. Children are normal. Okay, okay. Sometimes in the recessive, parents are normal, but still their child will show the trait. And sometimes parents are affected, still their children are not showing the trait. So it is skipping the generation, yes or no, Priyadarshan? They are skipping the generation. So that is a very important point, right? That is a very important point to rule out whether there is a dominant trait or the recessive trait. Okay, whether it is a dominant trait or the recessive trait. That's what you need to keep in your mind. And see, if you understand this now, right, then you have to see, you have to check few rules, and then then you can say whether it is autosomal or the allosome. So see, autosomal dominant trait. So here they have given the example of Huntington's disease. If you want, you can read about it. Okay, if you want, you can read about it. Now focus here, bacha. See, affected female, unaffected male. So in next generation, both the children are affected. Now here, one of the parents is affected, so in the next generation also you will find this trait. So no generation is skipped, no generation is skipped in their family history, no generation is skipped. So because it is autosomal dominant trait, it is not related to the sex chromosomes, it is not related to the XX and XY. So males and females are equally likely to be affected. Males and females are equally likely to be affected. Male and females are equally likely to be affected. Understood? Next. Affected offsprings have at least one affected parent. If offsprings are affected, definitely they will have at least one. There are chances that both are having the same trait or disorder, but at least one of them will be affected, right? So first of all, whenever you are going to solve this type of question, you have to rule out whether it's dominant or recessive. It will solve 50% of your, right? It is going to help you out like on the basis of that, you will solve 50% of the question. That's what I can say. Next step is to figure out whether it is autosomal or the sex-linked. Right? So first point clear, second point clear. Now, if affected offsprings are heterozygous, if only one parent is affected, it's fine now. Let's say that's the case. Let's say that's the case, or even that is the case. Okay. So if only one parent is affected, then definitely these affected individuals are having this. They are having this genotype. And if both are affected, let's say this one is also affected, both the parents are affected, then in that case they will be homozygous. They will be homozygous. It is not that important to remember, but still I will share the PDF in the Telegram group. You can refer to these points. Okay. So unaffected offsprings are homozygous recessive. Now it's a dominant trait. If their mother is affected now, so definitely mother will also pass the chromosome to the next generation, right? If this now here both are affected. Now let's say here 2 children, they are fine, 2 children are affected. So in that case, if you talk about their genotype now, definitely their genotype is homozygous recessive, because even if they will receive one, right? Not in the both the parents. Okay. See, here in this case, because it is a dominant trait now, if it is a dominant trait, there are chances that mother will pass its dominant allele. So even if they will receive single dominant allele, you know that character will express. So if it is a dominant trait, and you will see in a family that few children are normal, so it simply indicates that they are in homozygous recessive condition. Okay, I am not saying that you have to remember each and every point. You should know what is the meaning of dominant and recessive trait, and these are the points which help you to solve the questions quickly. If you will keep such points in your mind and you practice questions on the basis of pedigree analysis, then of course, right, these points will help you to solve it very quickly. Fine, you will get a clear answer. Okay, next is if one parent is heterozygous, about 1/2, basically 50% of the offspring will be affected, right? 50% of the offspring will be affected. Like if this is the scenario, or even if it is normal. So if this is the scenario, if affected individual is having heterozygous dominant, so 50% of the individuals will be affected. Obviously it is understood now. It is understood that mother will pass capital A to 50% of children, or 50% of offspring, of course. So trait doesn't skip generation. That's the most important point. So according to me, this one, they are the most important points, and the first one. So you can skip these 2 points as well if you want. Okay, if you want. Clear? Now move to autosomal recessive. So if it is a recessive, trait often skips generation. I will quote examples as well. See, carrier male and carrier female, that's how they are showing. Carrier, it means if they are carrier, it means they do not have that particular trait. Let's say they do not have that particular trait. Just say for an example, sickle cell anemia. let's say they are the carrier only. They are the carrier only. They have defected allele, right? They have one defected allele, but it is not expressing itself like this. So they are carrier. So see, basically parents are normal here. Parents are carrier means they are normal. Sickle cell anemia is the example. So see, parents are normal. Child is affected. Okay. Here also, parents are normal, but their child is affected. Here, parents are affected. Child is normal. So it means children are normal. So it means they skip generation. They skip generation. So males and females, see, whenever it is the autosomal trait, this is another important point to answer the question. Whenever you talk about the autosomal traits, whether it is dominant or recessive, males and females, there are equal chances of there to get affected. Okay, if it is sex-linked inheritance, then in that case, males, because if it is X chromosome, right, if it is X-linked inheritance, then males are more likely to be affected. Why? Because they have just one X chromosome and another Y. So if X chromosome is having defected allele, it will definitely express itself. But in the case of females, they have two X chromosomes. If one is having defective allele, another is having normal, females, they will compensate, no? Right? Right? So next is affected offspring often have unaffected parents. I told you, unaffected parents, they are, if they are unaffected parents of affected offspring, they are heterozygous. So affected offsprings are homozygous, right? So here definitely this is the genotype. This is the genotype. If it is recessive, now come to the sex-linked inheritance. So let's start with the sex-linked dominant. So see, look at it. It is sex-linked dominant. It is sex-linked dominant, right? So it is sex-linked what? Sex-linked dominant. In the sex-linked dominant, both are having equal chances of getting affected, right? Like if you will see this pedigree analysis, you will say male is affected, but in next generation females, and in next generation female male, both are affected. So clearly I can say this is X-linked dominant. Why? Because dominant means, in the case of females, I told you, even if two X chromosomes are there, even if one X chromosome is having the defected allele, it will express itself for sure. But if it is the X-linked recessive, then in the case of females, right, this is the scenario. If both the alleles are defected, then only they will express the trait, or else. But in the case of males, because one is X, another is Y, so even if they are having one defective allele, they will express it. So when it is X-linked dominant, both are having equal chances to get affected. Now, all the daughters, these points are important, bache. All daughters of an affected father are affected, but no sons. Can you explain it? All daughters of an affected father are affected. For sure they will be affected, because it is a dominant trait. It is a dominant trait, right? All the daughters of the affected father will be affected, but no sons. Let me tell you why. Because you know that males are having XY, females XX. Males are heterogametic. One gamete will contain X, another Y. But in the case of females, females are homogametic. Now, you know that if in the next generation, if you want a son, then mother will definitely give X chromosome, isn't it? If in the next generation, let's say if there is a couple, they want a son, so mother is going to form same type of gamete, so it doesn't matter. So father has to give Y chromosome only, right? Father is going to give Y chromosome, then only the son will be there, then only the male child will be there, right? So let's say father is having X-linked dominant trait. Father is affected with any X-linked dominant disorder. Okay, father is affected with any X-linked dominant disorder. So obviously, father will give Y to the son. So son is safe in that case. Son is safe in that case, because son will not receive that affected X from the father. But when you are talking about the daughter, daughters are going to receive one X chromosome from mother. Daughters are going to receive one X chromosome from mother, and they will receive another X chromosome from father. So if father is having X-linked dominant disorder, so definitely daughter will have that. daughter will have that for sure. For sure, daughter will have that. So that's why it is written, all daughters of an affected father are affected, but no sons. So that's a very important point if you want to figure out whether it is autosomal dominant or sex-linked dominant. Because in autosomal dominant, if father is affected, then son and daughter both can get affected. But if it is a sex-linked dominant trait, then if father is affected, then you will see in next generation, daughters are affected, not the son. Not even a single son in that case then. Right? Son will only be affected if mother is also affected. Right? Son will only be affected if mother is also affected. Clear? So, affected son always have affected mother, I told you, no? Affected son always have affected mother. If it is a X-linked, like let's say there is a cross, there is a pedigree analysis, there is this tree, right? Let's say in the exam, they are hiding this part. They are saying, see, it is a X-linked dominant trait. It is a X-linked dominant trait. The son is affected. Okay, so what about the mother? Or what about the parents? So obviously it is the possibility that definitely mother is affected. Okay, definitely the mother is affected. Clear? So 50% of the offspring of an affected mother will be affected. Okay, so affected daughters can have affected mother or father as well. And here also, because it is dominant, it will not skip generation. So if it is dominant, Vidisha, this point is fixed that it is not going to skip generation. So these points, these characteristic features, they are helpful or not? Tell me. Yes, they are helpful or not? They are helpful or not? Right? Yeah, bache, it's a good score, Anup. You should focus on class 11 syllabus as well. Now, if it is X-linked recessive, what is it? It is X-linked recessive. So see, male is affected, but female is unaffected. So in that case, this is the scenario, right? Because it is recessive, so males are more likely to be affected. See, males are affected more frequently than females. So this trait will never pass from father to son. Can you tell me why? Why? If it is the X-linked recessive trait, so father will always pass it to the daughter, and then that daughter will pass that trait to the son. But father cannot directly pass it to the son. So this is a kind of crisscross inheritance. So father will pass it to the daughter. Why? Because father will give daughter the X chromosome. So here son is safe. And then this daughter will give this X chromosome to the son. So it's kind of crisscross inheritance, isn't it? So here they are saying that affected sons are usually born to carrier mothers. Affected son are usually born to carrier mother. So what is the meaning here? So bache, best example here is your hemophilia. How many of you know about the hemophilia? You know na, hemophilia. It is the bleeder's disease. Right? What is it? It is the bleeder's disease. What? Hemophilia. Okay. Hemophilia, bleeder's disease. And it is also related to the royal family as well. I hope you know about it, royal disease. So you know that in our body, we have one mechanism, that is your blood clotting mechanism. And for this blood clotting mechanism, yes, we need certain proteins. For this blood clotting mechanism, definitely we need certain proteins like thrombin is required, fibrins are required, other factors are also there. So why this blood clotting mechanism is there in our body? That if we will get any injury, any cut, in that case, our blood should stop. Like let's say, even if I will get a minor cut, even if bleeding will continue for hours, obviously I'll die. So in that case, clotting is very important, right? In that case, clotting is very important. This is a very protective mechanism. Okay, this is a very protective mechanism. I told you already. Now the point here is, students, right? The point here is, okay, that in case if the clotting factors are not there, obviously blood even after a minor injury, blood loss will be there and that person can die. Okay, so such disease is hemophilia bleeder's disease. It can be because of two different factors, so we categorize it, right? It is also the Christmas disease as well. So males are affected here. Okay, here males are affected. You will see this disorder in the case of males, and females are the carriers only. Females are carriers only. They are affected and they will remain unaffected, but they are carriers. What is the meaning of being a carrier? Let me explain you. Now see, hemophilia is a X-linked recessive disorder. What type of disorder it is? X-linked recessive. So if it is X-linked recessive, so see, females are having two X chromosomes. If both the chromosomes are having defected allele, then only females are affected, yes or no? Then only females are affected if both the X chromosomes are having defected allele. But this is not possible. This is not possible. It's a lethal combination. It's a lethal combination. I told you to read about the lethal genes. In the lethal genes, instead of 3:1, the ratio is 2:1, because one combination, it doesn't exist. In the case of lethal genes, the ratio is 2:1. Why it is not 3:1? Because lethal combination will not exist. So here, when it is hemophilia, okay, it's a X-linked recessive disorder. So in that case, both the X chromosomes should have defected allele, then only female will become hemophilic. It is not possible. Such type of zygote will not survive, it will die. So females are never ever affected with hemophilia because it is a lethal combination. Now, let's say one X is having defected allele, but another X is normal. So in that case, females are not having hemophilia. Females are just the carriers. Carriers means they have that affected allele, but they can pass it to the next generation. They are not affected, but they can pass it to the next generation. So hemophilia is the best example there. So that's why we say if males are affected, definitely, right, males are affected more frequently than females. So trait will never pass from father to son. It will pass from father to daughter and then daughter to the son, like father to grandson it will pass. Affected sons are usually born to carrier mother. So here the mother is carrier. So if carrier mother will give its X chromosome to the son will get the disease because father will give Y. Okay, so 50% chances are there na, 50-50 chances are there na. So if mother will pass defected allele, defected X chromosome to the daughter will be the carrier. But if mother will pass defected X chromosome to the son will be diseased. Clear? So about 50% of the sons of a carrier mother will be affected. 50% of the sons of the carrier mother will be affected. Clear? So all daughters of affected male, if father is affected, definitely all daughters are going to be. See, father is affected, mother is normal. Mother is normal means this condition, father is affected. So definitely female child will be carrier. Female child will be carrier in that case. But if mother is the carrier, chalo, I'll give you one cross, try to solve it. I'll give you one question here, try to solve it. If let's say this is the scenario that father is affected, mother is the carrier, then what is going to happen? Please tell me what is the possibility here. Yes, Ahiti, what is the possibility here? Tell me what is the possibility here. Please write it. Please be active in the class. In that case, see, this combination will not exist. It's a lethal combination, means death will occur. So the ratio is 2:1. And here you will see daughter is a carrier, son diseased, son is normal. So it means if mother is the carrier and the father is diseased, so in that case, right, I just told you na, that point was explaining na, that in that case, if mother is carrier and the father is affected, 50% of the sons, they'll be affected and 50% is normal. That's the meaning, right? So if you ever get confused, you can draw the Punnett square and then you can answer it, okay? Then you can answer it. Clear, bache? Clear, bache? So that's it. That's all, bache. So I know it is not possible for you to remember each and every point in, you know, in just one class. But after the class, of course, you can revise it, isn't it? You can revise it. And then if you will revise it, if you will practice question on the basis of that, it will be easy for you. Now, let's read this part from NCERT and then we will start the PYQ practice, MCQ practice, especially the practice of your pedigree analysis based question. That's what we have to do. Ready? Yes. Are you all ready? Okay, let's go. So that's what we were studying, that as you have studied in this chapter, each and every feature in any organism is controlled by one of the another genes located on the DNA present in the chromosome. So DNA is the carrier of genetic information. Of course, we know that. Of course, we know that. Right? So it is the DNA that will transmit from one generation to another without any change or alteration. Right? So however changes or alteration they do occur occasionally, right? And we used to call it as mutation. So now you know what are mutations, right? Right? That alterations in the DNA structure in the genes. So number of disorders in human beings, they are found associated with the inheritance of changed or altered genes. So if there are mutations that can cause these disorders. Is that clear? Is that clear? Yes, everyone. Is that clear? So Mendelian disorder. Let's start with what? Mendelian disorder. So yes, in the paper they even ask which disorders are the Mendelian one or the chromosomal one. So you should know about it, right? You should know about it. So now what are we going to start? We are going to start Mendelian disorders first. So broadly genetic, see, now you know the mutations, okay? You will understand it in a very easy manner. Fine. So genetic disorders may be grouped into two categories, Mendelian and chromosomal. Which disorders are Mendelian one, bache? They are mainly determined by alteration or mutation in the single gene. So it is related with the change in the single gene. This point is important, Sahiti. This point is very important. You should know which disorders are Mendelian, which disorders are chromosomal. So if there is a change in the single gene, it will be your Mendelian disorder. It will be your Mendelian disorder. So they will transmit to the offspring on the same lines as we have studied in the principles of inheritance. Now, what is the meaning of this? You have studied the laws of inheritance, so accordingly they will transmit, right? That autosomal one, allosomal, right? A sex-linked chromosome, autosomal inheritance. So this is what they are going to follow. So pattern of inheritance of such disorders, it can be traced by the pedigree analysis. Now, everything is making sense to you or not? Right? So in genetics, in human physiology, in plant physiology, you need a teacher. You need a good teacher that can make NCERT easy for you. Right? The teachers who can make, those who are making NCERT easy for you, that are good for you. Ultimately, you need to read this book, you need to understand this book and never try to cram it, understand it. If you will understand the meaning of each and every line there, each and every word there, then it's very easy to get 360 out of 360, trust me. It's very easy to get 360 out of 360. So these disorders are transmitted to the offspring on the same lines as we have studied in principles of inheritance. So pattern of inheritance of such Mendelian disorder, it can be traced in a family by pedigree. So Mendelian disorder, if there is a change in a, or if there is alteration in a single gene. Any doubt? Any doubt? So most common disorders are, see, examples can be asked. Hemophilia, I explained to you. Cystic fibrosis, this is what we need to discuss yet. And then comes the sickle cell anemia and the color blindness as well. Phenylketonuria as well. Thalassemia as well. So another important MCQ. If I will be the examiner, I'll give you the options. I'll ask you which one is the Mendelian disorder, which one is the chromosomal one. So that's what you need to know. Hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia. And you know another question can be, like I told you, hemophilia, bleeder's disease. It is the example of autosomal recessive disorder. Sorry, It is the example of X-linked recessive disorder. Okay, and now if you are talking about the phenylketonuria, I explained phenylketonuria to you now. In the pleiotropy also we have discussed it. So it is the example of autosomal recessive disorder. It is the example of autosomal recessive disorder. So you need to focus on such things as well. Which one is autosomal recessive or X-linked recessive? Okay. So by pedigree analysis, we can check that, bache. So trait may also be linked to the sex chromosome, like your hemophilia, right, bache, right? So this is given here. Okay. So see, one is autosomal dominant for myotonic dystrophy. So myotonic dystrophy, this can be asked as a question. So myotonic dystrophy, this is the example of an autosomal dominant trait. So another important homework. But if you are following me sincerely, na, right, and the homework that I'm giving you, right, if you even follow that sincerely, trust me, no one can stop you to score 360 out of 360, because even if I'm taking a lecture for 3 hours, for 4 hours, or for 5 hours, these are the detailed lectures, right? We are not going to start one shots. Yes, that one shots where, you know, we will skip some things and we will directly start from advanced level. No, right now if we are taking the one shots, in that one shots we are explaining everything in detail. So they are detailed one shot. Still you have time. In the month of January we will start that one shot in which, you know, we will start from advanced level only. So as of now till December, whatever we are, you know, teaching you, please start making the notes, especially in the biology. If you will follow these classes, definitely you will get 360 out of 360. Okay, but you have to practice the questions as well. Okay, so myotonic dystrophy, it is the example of autosomal dominant trait, and it is your homework. You will read about it. So just look at this. No generation is skipped. If mother is affected here, son is also affected, daughter is also affected, right? Both the parents are normal, so their children are also normal. Parents, one of the parents affected, in next generation you will also see the trait, right? So here you cannot say that daughters are more likely to be affected or sons are not. See, no doubt son is affected, daughters are affected, plus son is normal as well, daughter is normal as well, right? Daughter is normal as well. So that's why it is autosomal. That's why it is autosomal. Clear? And next is, and yes, how can you say that whether it is X-linked or not? Whether it is X-linked or not? How can you say that? How can you say that? Like, how can you differentiate in between autosomal dominant and X-linked dominant? Vidisha, how can you differentiate in between autosomal dominant and X-linked dominant? Yes? Okay, read about it. You will solve question, you will get to know. Now see, parents are normal, children are affected. Parents are normal, children are affected. So definitely generations are skipped. It is recessive. So it is this pedigree analysis. This pedigree analysis is the representative of sickle cell anemia. Clear? Sickle cell anemia. So now let's start with the color blindness. So what do you know about? What do you know about the color blindness? Tell me quickly. What do you know about the color blindness? Only one topic is left now, mutagens. Okay, so first of all, tell me about the color blindness. Tell me. See, when it is the color blindness, again, it is also X-linked recessive disorder. Fine. Here, if you talk about the males, right? See, 8% of males are affected and just 0.4% of females. So when it is color blindness, that individual cannot differentiate red and green color. That individual cannot differentiate red and green color, right? Red and green color. Are you getting it? Are you getting it? Yes. So it is due to the mutation in certain genes which are present on X chromosome. So here 8% of males and 0.4% females are affected. So a very common mistake that students used to make here, they think that color blindness is also there in the case of males only and females are not affected. It's not like that. It is affecting males as well. It is affecting females as well. It is the hemophilia which will be there in the males only. It will not be there in the females because it is a lethal combination. Clear, bache? So this is because the genes led to red-green color, they are present on X chromosome. So they are saying males have only one X chromosome, females have two. So son of a woman who carries the gene has a 50% chance of being colorblind, right? I'm repeating this again. The son of a woman who carries the gene has a 50% chance of being colorblind. In the case of hemophilia also I explained the same thing. Like if female is carrier, right? Female is carrier. Let's say father is normal. Father is normal, female is carrier. Still, let me draw this. If see female is carrier and the father is normal, see 50% chances that son will be colorblind, and even father is normal and the mother is a carrier, then there again 50% chances are there that daughter will be the carrier. So in the case of females, if this is the case, like both the X chromosomes are having this defected allele, then only females will also be affected. That's why the chances here are less. Clear? It is 0.4%. Okay, so mother is not herself colorblind if mother is a carrier, right? So because another X chromosome is having the normal allele, okay, so daughter will not normally be colorblind unless her mother is a carrier and the father is a colorblind. So is that clear? Color blindness done, Mendelian disorder. Next is hemophilia. I already explained it. It is having the type, right? It is dependent on two different factors. That's again your homework. I will keep one lecture where we will practice the questions of these disorders only, because for sure you will get 2 questions in the final NEET paper from this part. So hemophilia again, sex-linked recessive disease. So please make the notes. Okay, in that notes, first line should be color blindness, X-linked recessive. Hemophilia, X-linked recessive. You should know about it. So again here, bache, same story is there. The transmission from unaffected carrier female to some of the male progeny will be there. In this case, single protein, which is the part of cascade of protein involved in blood clotting. You know, blood clotting is not a single step process. It's a multi-step process. It's a cascade, basically, one after another. Like you know that, first of all, what will be there? Thromboplastin. Thromboplastin will be released by platelets. Thromboplastin will be released by your injured blood vessel. That thromboplastin will act on prothrombin, will make it thrombin. That thromboplastin will also help in inactivation of heparin. We need calcium for blood clotting as well. So once thrombin is there, right, then what will happen? Think about it. So basically, first thing, thromboplastin needs to be there, which will activate your enzymes also. Okay, so it's a cascade of process. It is not a single, right? It is not a one-step process. Fine. It is not a one-step process. Okay. One after another. There is a proper sequence that needs to be followed. Okay, there is a proper sequence that needs to be followed. One factor will activate the another factor. One factor is going to activate the another factor. Clear? So that's what you need to study. Okay, that's what you need to study. Done? So here, in the case of males, such factors will not form. That's the point. So next is, due to this affected individual will have continuous bleeding even if there is a minor injury. So heterozygous female for hemophilia may transmit the disease to son. So it is all same. The inheritance is same like color blindness, but you have to remember about the lethal combination. So family pedigree of Queen Victoria, right? Family pedigree of Queen Victoria shows a number of hemophilic descendants as she was the carrier of the disease. So that's why it's a royal disease also. Royal family was having this. Okay, royal family was having this. Next is sickle cell anemia. So I am explaining the sickle cell anemia again. So it is also showing the pleiotropy. Pleiotropy, if you remember. Remember what is pleiotropy? Pleiotropy is the multiple phenotypic effects of a gene. Okay, it is the multiple phenotypic effects of a gene. Means a single gene is controlling multiple characters, right? Single gene, like it is affecting the shape of the RBC, it is causing harm on the liver, also the eye disease also. That's how it is related. That's how it is related. So when you talk about the sickle cell anemia, first thing that you need to remember it is autosomal recessive, right? Autosomal recessive trait. It is not linked with the X chromosome. It is related with the autosomal. So males and females, they have equal chances to get affected. Now you know that it also shows what? Codominance. This is, remember this possibility, right? One allele is affected, another allele is, right? One allele is affected, another allele is normal. So in that case, that person will survive. If this is the scenario, like both the alleles are for sickle cell anemia, the person can also die. Okay, the person can also die. So it is autosomal sex-linked disorder. Now you know that what is actually happening. I'm repeating this again. Please take a note of it. So you know that we have hemoglobin. So hemoglobin is a quaternary protein. What type of protein it is, bache? It's a quaternary protein. It is having 4 polypeptide chains. How many chains are there? 4 polypeptide chains are there, right? And globin is the protein part, heme is the pigment, and it will have it contains Fe2. So this is Fe2 positive, Fe2 positive, Fe2 positive, Fe2 positive. Ferrous it is, na, right? So it is going to bind with the oxygen. That's what we know. But now hemoglobin, right? Quaternary protein it is. So it is having, adult hemoglobin is having 2 alpha chains. It is having 2 beta chains. It is having 2 alpha chains. It is having 2 beta chains. Clear, bache? 2 alpha chains, 2 beta chains. Now in beta chain at 6th position, because ultimately it's a protein. So it is made up of amino acids. It's a protein. It is made up of amino acids. Okay. It is made up of what? It is made up of amino acids. So at 6th position, see 1, 2, 3, 4, 5, 6. Normally at 6th position, you used to get your GAG. Now, why am I writing it like this? GAG. Actually, it's a triplet base codon which is forming glutamic acid. Okay. Right? Amino acids are formed. They are formed by these codons, by these nitrogenous bases. Right? In the genetic code, we study this, that there is a relation in the mRNA and in that amino acid, in the proteins. Proteins are what? Polymers of amino acids. mRNA is the polymer of nucleotide, but there is a correlation, there is a relation in between them. So these three, right? These 3 nucleotides, they form one amino acid. So GAG, G-A-G. Guanine, adenine, guanine, it forms glutamic acid. So normally it is there. So after mutation, here GAG will become GUG. This is how I remember. GAG will become GUG. So now when it is GUG, see, there is a change of only one base. There is a change of only one base. Only one base. So that's why we call it as point mutation. And it's the example of substitution. It is a point mutation. It is the example of substitution. It is a point mutation. It is the example of substitution. So glutamic acid will become valine. So when valine is present here, now your RBC is not normal. Your RBC will become sickle shaped. Right? So basically the sickle cell anemia is also a blessing in disguise. So the person having sickle cell anemia, mainly in Africans we see this. Right? Malarial parasite, they will not get affected with malaria. If a person is having sickle cell anemia, that person will not get affected with malaria. Why? Because malarial parasite cannot survive in sickle shaped RBCs. So that's why it is blessing in disguise. Okay, so sickle cell anemia, I told you about it. It's an autosomal linked recessive trait. It is transmitted from parents to offspring when both the partners are carrier, right? Both are heterozygous, like this is the condition. Okay, okay. So you know na, the disease is controlled by single pair of allele, HBA and HBS. So out of 3 possible genotypes, only homozygous individual for sickle cell anemia shows the diseased phenotype. Heterozygous, they appear apparently unaffected. They are the carrier. So I told you na, codominance is there. Codominance is there, right? The person will be normal, hai na? Yes, bache. Yes or no? Yes or no? Okay. And do you know where the gene for the synthesis of this beta chain is located? Do you know that on which chromosome? On which chromosome? So actually on chromosome number 11, this is also a previous year question. On chromosome 11, gene for forming beta chain is located. Okay, it is located there. Clear? It is located there. Fine, bache. So that's it. So this defect is caused by the substitution of glutamic acid and it will form valine. So this diagram can also be asked in the paper. Fine. So normal A gene, GAG, and this, because it is a gene, DNA, so CTC is written. So you know that gene is forming mRNA, means transcription, then translation is going on. So here on mRNA you will get GAG. So if GAG is there, glutamic acid. So glutamic acid is also denoted like this, GLU. So GAG means the triplet base codon, and GLU, if I'm writing it like this, means glutamic acid. So if it is sickle cell gene, here it will be GUG, you will get valine. So bache, in the paper they can ask you this part as well, right? They can ask you this sequence. So valine, histidine, leucine, threonine, proline, glutamic acid and glutamic acid is there. So that's what you need to remember as well. That is also important, right? That is also important. Clear? Clear, clear, clear. So this is, as I said, it is the substitution of amino acid, right? Due to the single base substitution at 6 codon. So mutant hemoglobin will undergo polymerization under low oxygen tension, causing the RBC to form biconcave disc to elongated sickle cell structure. Simple. Next is phenylketonuria, again autosomal inborn error of metabolism. Remember, in that case, what is the case? What is the scenario? Phenylalanine hydroxylase will not form. Vidisha, even we have discussed that, yes? Even we have discussed that. Yes or no? Yes or no? Phenylketonuria, do you remember phenylketonuria? Yes. So it is an inborn error of metabolism. So here this gene is present on chromosome number 12. Chromosome number 12. Okay. So it is autosomal recessive. So affected individual is not having the enzyme. Which enzyme is absent here? PAH, phenylalanine hydroxylase. Okay. Phenylalanine hydroxylase. So this enzyme, phenylalanine hydroxylase, is absent. So this enzyme is absent. So phenylalanine will accumulate. Mental retardation can occur. Skin pigmentation can occur, because this enzyme also helps the conversion of phenylalanine to tyrosine. Okay, okay. I have explained it earlier, bache. Right. So this enzyme will help us to convert your phenylalanine to tyrosine. So this tyrosine is very important for the formation of the skin pigment melanin. No tyrosine means no melanin. No tyrosine means no melanin. So skin pigmentation will be there. See, phenylalanine into tyrosine. So as a result of phenylalanine is accumulated, because it is not getting converted into phenylpyruvic and its other derivatives. So accumulation of these in brain can result in mental retardation. So it can also excreted through urine because of its poor absorption by kidney. So this is important. So Aditya is asking, ma'am, what type of disorder it is? Sickle cell anemia. Quantitative or qualitative? Bache, here it is related to the quality, right? Your RBC shape is changed. It is a qualitative disorder. When you will talk about the thalassemia, there the amount of globin is less. So it is quantitative disorder. Okay, then it will be quantitative disorder. Fine. Yes, we are about to finish this chapter and then we will solve the MCQs. So see, next is also thalassemia, autosomal linked recessive blood disease. So it is also transmitted from parents to offspring. Defect could be due to either mutation. See, I told you na, deletion can occur, which ultimately result in the reduced rate of synthesis of one of the globin chains. Right? See, bache, Aditya, you will get the answer of your question. You will get the answer of your question here, that the mutation, or you can say that deletion, because of that what is happening? Reduced rate of synthesis of one of the globin chain. It can be alpha or beta, right? So on the basis of that, even you have different types of thalassemia. Minor, major thalassemia is also there. So see, so that make up the hemoglobin. So of course it is related to the quantity. Fine bache, fine bache, thanks Nishat. Right, it is related to the quantity. It is related to the quantity. So you should know on which gene, chromosome, which gene is present. Okay, 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 okay. Just I told you about the alpha chain and the beta chain, right? For the alpha chain, for the synthesis of alpha chain, chromosome number 16, it is having the gene for the formation of both the alpha chains. And bache, when it comes to the beta chain, yes, when it comes to the beta chain, I told you for beta chain it is on chromosome number 11. You need to remember that, right? For the synthesis of alpha chain, the gene is present on chromosome number 16, but for the beta chain, the gene is present on chromosome number 11. So if it is alpha thalassemia, means alpha chains are deficient. Fine. Alpha chains are deficient. I told you na, hemoglobin is having two chains, 2 alpha, 2 beta. Alpha thalassemia means alpha chains are not there. Beta thalassemia, beta chains are not there. So this is very important. This is very, very, very important. Chromosome 16, alpha chains, right? Chromosome 16, alpha chains. Chromosome 11, beta chains. Okay? Beta chains. Fine. Fine. So abnormal hemoglobin is formed. Quantity is less. Right? Right. So see, in alpha thalassemia, production of alpha globin chain is affected. In beta, production of beta globin chain. So alpha thalassemia is controlled by two closely linked genes, HBA1 and HBA2, located on chromosome 16. So this is the gist of the topic. That's important. This is the gist. Okay. And it is observed due to the mutation or deletion of one or of the 4 genes. So total 4 genes are there, right? If there is a deletion of even any of the gene, right, it can result in this thalassemia. So more genes affected, the less alpha globin will form. And in the beta thalassemia, it is controlled by single gene HBB on chromosome 11. This information is important. Vidisha, this information is important. Fine. So thalassemia differs from sickle cell anemia. Sickle cell anemia is quantitative problem, and because thalassemia is quantitative problem, sickle cell anemia is qualitative. Another important MCQ, right? So here, bache, actually for these disorders, na, NCERT reading is very important. NCERT reading is very important here. Next is the chromosomal disorders. Wow, we are about to finish it and then we are about to start the pedigree analysis. Excited? After finishing it, we will take another 10 minutes to finish it, and then after that, we'll start what? We will start your pedigree analysis, MCQs of pedigree analysis. So I hope you guys are excited for that. Seriously, I hope you people are excited for it. Okay, so see, you know about chromosomal disorders. So if there is a failure of segregation of chromatids, if chromatids are not getting separated properly, aneuploidy, right? So aneuploidy may, you know what you have to study. Trisomy, when there is the addition of a particular chromosome. Monosomy, when there is the deletion of a particular chromosome. Okay, so for example, Down's syndrome, it is the resultant gain of extra copy of chromosome 21. Let me show you this. But see, this is the karyotype, right? This is the karyotype. Do you know what is the meaning of a karyotype? Anyone here in the class? Yes, Shiva Kumar. What is the meaning of karyotype? What is the meaning of karyotype? Anyone in the class? Karyotype. Yes, it is actually the appearance. It is the number and appearance of the chromosome present in a cell. Yes or no? Saki, this is what karyotype is now. It is actually that representation of the complete set of chromosomes which are present in a human being. Yes or no? Right? Right? Yes or no? Yes or no? So let's say if a person is having Down's syndrome. Down syndrome means trisomy 21. So trisomy 21 means one extra copy of your 21st chromosome, right? Because segregation is not there, non-disjunction. So look at the karyotype here. 1, 2, 3, 4. Come to this 21. So here, yep, this is having one extra copy, right? Just check your NCERT. It is not just one. 3 chromosomes are there actually. It should be 1 na. Ideally, it should be 2 na. Chromosome number 21 will be there in the pair na. It will be there in a pair na. One from mother, one from father. So we have one more extra. So that's why it is trisomy 21. Right? So we even have another examples also. Just say you have trisomy 18 also, trisomy 13 also. Right? Trisomy 18 is also there. Trisomy 13 is there. So Subhash Bache, if it is the trisomy 18, right? If it is the trisomy 18 or 13, so it will cause some another disorders. Just say trisomy 18 is also known as Edward syndrome. Right? If you talk about the trisomy 18, means you have extra copy of 18th chromosome, then you will call it as Edwards syndrome. Okay. One is trisomy of 13th chromosome. That is your Petau's syndrome. Petau's syndrome. What's that? That is your Petau's syndrome. So trisomy means one extra copy. So this is aneuploidy. So just now in this lecture we have discussed this na, genomatic mutation, chromosomal aberration. So see, now everything is relatable, isn't it? Isn't it? So trisomy 21 means Down syndrome. So see, flat back of head, broad flat face, many loops on the fingertips, big and wrinkled tongue will be there, palm crease will be there, and congenital heart disease. What is the meaning of congenital? Congenital means by birth, right? By birth, heart diseases will be there. So when you know in the mothers, when females they conceive in the later ages like 30 plus or sometimes mostly 35 plus. So in that case, the chromosomal disjunction at the time of gamete formation, it gets affected. So there are chances that in such cases, child can have Down syndrome. Okay, child can have Down syndrome. That is the case then. So now just look at it, Turner's syndrome. Turner syndrome is also the example of your aneuploidy. But here it is due to the loss. So means it is because of the monosomy. It is because of the monosomy. Monosomy is 2n minus 1. And here in Turner syndrome, there is a loss of X chromosome. Right? There is a loss of X chromosome. Clear, bache? So failure of cytokinesis after telophase stage of cell division, it results in an increase in whole set of chromosome, that will be polyploidy. So aneuploidy, polyploidy, difference clear, disorders clear. Yes. Difference clear, disorders clear. So let's start with the Down syndrome. So the total number of chromosome in normal human is 46, 23 pairs, 22 pairs are autosomes, 1 pair is sex chromosome, allosomes, right? So sometimes, though rarely, either an additional copy of chromosome may be included. Okay, okay, or they can lack even. So these situations are trisomy or monosomy. So you know what is the meaning of trisomy and monosomy. So Down syndrome, Turner syndrome, Klinefelter syndrome, these are the major chromosomal disorders. So let's start with the Down syndrome. So you know that additional copy of chromosome number 21, first discovered by Langdon Down. That's why it is Down syndrome. So affected individual is having short stature with small round head, furrowed tongue, partially open mouth, palm is broad, right, and palm crease are present. So physical, psychomotor and mental development is retarded. So you should even mark in which disorder mental development is retarded or not. Fine. So symptoms are important, and you know that it is, you have to mug up these points. Then Klinefelters. Again, it is also the example of trisomy. But here there is the trisomy of the X chromosome. Trisomy means one extra copy of X chromosome. Okay, so normally if it is XY, it can be XXY also, right? So femaleness will be more, female-like characters will be more. So genetic disorder in which there is an additional copy of X chromosome resulting in the karyotype 47. So how can you write it? 44 plus XXY, 45, 46, 47. Isn't it? Isn't it? Right? So in such an individual, overall masculine development will be there, but feminine development is also there. So do you remember gynecomastia? In the chapter human reproduction, we have discussed it, na? Gynecomastia. So gynecomastia is because of hormonal imbalance. Here in this case, males, in the case of males, breasts are developed. I'm not saying they are functional, but the growth of breast is there in the case of males. So gynecomastia. Okay, gynecomastia. It is because of hormonal imbalance. Why? Because in the male, extra copy of X chromosome is there, femaleness will be there, hormonal imbalance will be there. So such males are sterile, right? Such males are sterile. So Turner syndrome is the example of monosomy of X chromosome. So 45 chromosomes are there in totality. So this is how you can write it. One X chromosome is missing. Okay. Okay, so such females are also sterile. Ovaries are rudimentary. Rudimentary means some they are not fully developed. There remain some remaining parts are present there. Okay, okay. So secondary sexual characters like breast development and all such things are absent. So this is about the chapter. So let's start with the MCQ practice, and here we have our first question. So let's start it. So if there is any doubt, do let me know, students. If there is any doubt, do let me know. That is Shiva Priya. In that case, different possibilities are there. That's a very detailed topic. Let's not discuss it now. Okay, so let's start it. So now you tell me, bache. So I want your active participation, please. Fine, I want your active participation. So quickly tell me this. So trait shown in the pedigree chart, it is most likely, yes, it is most likely, what is it? Is it autosomal recessive? Is it autosomal dominant? And it is sex-linked recessive or sex-linked dominant. So what should be the answer there? What should be the answer? What should be the answer? And I'll ask for the explanation as well. It should not, you should not cram it. See, parents are normal, but their children are having disease. Okay, so definitely it is recessive. So you can cancel this option, you can cancel this option. So how will you decide it is X-linked recessive or autosomal recessive? Male and female both are affected, right? So if it will be the X-linked recessive, so males are more likely to affect it. So that's why we are saying it is autosomal recessive. Next question, this one. Now, please answer it to me. This one. This one. Again, it's easy. Quick. Yes. See, bache, first of all, observe one thing. You need time or should I explain it? Bache, see, look at it. Parents are affected. Okay, you will see it in this generation. Here, parent is affected, but the children are normal. So it cannot be dominant. It cannot be dominant. It's recessive. It's recessive for sure. See, parents are normal, children are affected. Definitely it is recessive. It is recessive. So cancel this option. Why are you saying D? Now, how can we say it is autosomal recessive or X-linked recessive? So look at it, right? Males are more affected, so it should be X-linked recessive. It should be X-linked recessive. Next, this one. Yes, this one. Answer it. Answer it. Yes, bache, answer it. See, it's very simple. You can go with the elimination method. Pedigree chart is wrong as this is not possible. No, it's completely fine. Okay. Inheritance of condition like phenylketonuria as an autosomal recessive disease. Wait, they are saying inheritance of recessive sex-linked disease like hemophilia. If it is hemophilia, then this case is not possible. So definitely it is not the answer. Inheritance of a sex-linked inborn metabolism. Phenylketonuria is not sex-linked. It is autosomal recessive. That's why B is the correct answer. That's why B is the correct answer. So next question. Study the pedigree chart of a family showing the inheritance of myotonic dystrophy, the trait. This is a direct question from NCERT. They are clearly specifying its myotonic dystrophy. So if it is myotonic dystrophy, it is autosomal dominant. If it is myotonic dystrophy, it is Shiva Kumar, autosomal dominant. Okay, next trait. Yes, next trait. Yeah, we have so many questions. Next traits. Given below is a pedigree chart of a family with 5 children. It shows the inheritance of attached earlobes as opposed to the free ones. The squares represent male individual and circles the female. Which one of the following conclusion drawn is correct? Yes. Even this is easy. The parents are homozygous. See, the trait is Y-linked. If it is Y-linked, see, Y-linked, if trait is Y-linked means all the males will be affected. But here it is not like that. So this is not the possibility. Parents are homozygous dominant. If parents are homozygous dominant, definitely their children should show the disease. All of them should show the disease. And it is not a dominant trait because generations are skipped. Cancel it. Okay. So parents are homozygous recessive. If they are homozygous recessive for a trait, they should show the character. So we are left with D. So this is the elimination method that you can use here. Okay. This is the elimination method that you can use here. Next question. Given below is a pedigree chart showing the inheritance of certain X-linked trait in humans. So here the answer should be yes. Is it a dominant one or the recessive one? Is it a dominant one or the recessive one? Again, it is also very easy, bache. No generation is skipped. And it cannot be Y-linked because females are also affected. Simple. Okay, it's very, very, very simple. So few more questions are there, right? That you will solve later after the class. Fine, that you are going to solve later after the class. Okay, so now you know what you have to do. You know what you have to do. I have posted a strategy video. Go and check it out. Fine, go and check it out. Accordingly, plan a schedule. And in the comment section, I want to see that comment that yes, ma'am, we are going to follow it. Okay, and few questions are still pending that you will solve. Okay, we'll share the PDF in the Telegram group. You are going to solve it. So some another day we will keep the one shot as well. For this chapter also we are going to keep the one shot ad as well. Okay, so as of now just revise this chapter. Even in our classes we have included that NCERT snips, so I don't think that you will find it difficult to understand NCERT. Okay, Shiva, I have shared all the PDFs. Okay, still I will check if there is any. I will share it in the Telegram group. And moreover, you know, your Avengers batch is one of the best batches where human physiology is going on, and soon we are going to start class 12 syllabus. So I am going to start your class 12 syllabus. So even if you are in class 12, you are preparing for the boards as well as for the NEET, this course is going to help you out, right? You will complete your syllabus in next 2 months. Okay, so you'll be ready for the boards as well, and then you can also use the time for the revision of the, right, for the revision of what? For the revision of your class 11 syllabus. Now you know what you have to do. What you have to do now, you people need to put a comment in the comment section and I will see the homework. Molecular basis of inheritance will start from Tuesday. Okay, it will start from Tuesday. Okay, so check out the strategy video, make the notes, utilize your Sunday wisely. Okay, we'll try to post something good for you so that you can feel motivated and good. So next chapter is molecular basis of inheritance. In 1 week we will finish it. Parallelly we will also complete body fluid and circulation as well, and next week we will go for the evolution. So we are covering most important units, so kindly be consistent in the lectures. Okay, so thank you, stay tuned, bye bye.